Alright, in this relatively short video, we're going to talk about how DNA replication can happen relatively quickly, despite the fact that there are so many base pairs that need to be replicated in our genome. And so just a quick reminder about semiconservative replication, because that's so important. We have our two strands of DNA, and each strand is linked by phosphodiester linkage, covalent bonds linking that strand together, and this strand the same. And then the two strands are held together by hydrogen bonds. And we know that hydrogen bonds are weaker than covalent bonds, so that can be unzipped. Those hydrogen bonds can be broken, and the two strands can be separated. So here we have this strand right here is over here. This strand right here is over here. And now each of these strands is acting as a template because we have base pairing that comes in. So we know that to this G is going to bind a C because of hydrogen bonding. And to this C is going to bind a G and so forth. So basically, the new strands get made from 5' to 3'. And we're going to learn all about DNA polymerase that links these covalent strands together. But again, it's important to remember that the base pairing is and hydrogen bonds between them is what causes T to come in when the template showed in A and so forth. All right, so that's semi-conservative replication. The two original strands are separated. Each of those acts as a template for the new strand, which grows 5 to 3. So where this starts is called a replication fork. So this is a point right in the middle of the DNA that starts to unwind. And it starts to unwind, and the replication is going to occur in both directions. We call that bidirectional synthesis, which is what our focus will be on today. So this would have originally formed this little bubble here, and then that bubble extends as the new strand gets added in both directions. And so that bubble is going to keep going, keep going. That's the replication fork. And we can see that here. This is a electron micrograph, and we can see the DNA. This is the DNA with the original double helix. There's the DNA with the original double helix. This would have started to form its bubble, its replication fork here, and you can see that now we have two strands, and the replication fork is going to be growing in this direction as that single double helix gets replicated and becomes two double helices, and same on this end over here. So this replication fork is expanding in both directions as the DNA gets copied from one double helix to two double helices. All right, so step 2, after the replication fork is opened, we need an RNA primer. And we're going to talk about why we need an RNA primer in Wednesday's class. And it's because of the restrictions for DNA polymerase, which is the enzyme we'll talk a lot about. But we create an RNA primer, right? So the replication fork would be going this way as the original DNA is unzipped and to act as a template. So we have a primers, all right? And then each strand is acting as a template. So if we have an A here, remember it's hydrogen bonding, it's complementary base pairing. That means a T is going to bind here. Over here we would have a G coming in to bind to that C. So that base pairing occurs, and then DNA polymerase is going to catalyze the phosphodiester linkage between this T here and the C that's in its strand. So this strand is growing. The new DNA nucleotides are coming in based on base pairing, complementary base pairing, and then DNA polymerase is catalyzing the linkage, the phosphodiester linkage, to the growing strand. And so here we can see the G coming in, and that'll be linked by DNA polymerase to this T. Here we see the T coming in, that'll be linked by DNA polymerase to the C. And there's a reason we kind of show this growth in two different directions. We're going to talk about that today as well. This is one of the complications of bidirectional synthesis. So prokaryotes have a circular genome, single chromosome, circular chromosome. So here's the origin of replication. You can see that bubble starts to expand, and now we're getting two strands. All right, so this is basically becoming copied, and the replication fork will be growing in this direction. You can see it expanding, continuing to expand until eventually we have two full circular chromosomes. So that can happen because most prokaryotes have about 4 million base pairs. However, humans have 3 billion base pairs that need to be copied. To put this into perspective, if you were typing 100 words per minute, which is actually quite fast, and let's assume the average word has 6 letters, that means you're typing 600 base pairs per minute. It would still take you 3,472 days to copy the entire genome. That's about 9.5 years. Your cell does this in a matter of minutes. So how is it able to copy these 3 billion base pairs not only so rapidly, but so perfectly without mistakes. How would you make it more rapid? Let's think about this. So, which will be faster? Lots of replication forks or one replication fork? Well, hopefully it's quite obvious that if you are doing replication at multiple places at the same time with lots of replication forks, that's going to be faster. If you have many nucleotide monomers available, or if you only have a few nucleotide monomers available, well, obviously it's going to be faster if there's a lot of nucleotide monomers available, because then you have one ready to bind as soon as the template's opened, and you're not having to wait around for that. And then, is it going to be faster to not have an enzyme, or to have an enzyme that links that phosphodiester linkage really quickly? Because we know enzymes catalyze reactions and make them faster. Well, obviously, having an enzyme that works very rapidly for that covalent linkage is going to be faster, and that is DNA polymerase. Okay. So, I think I'll stop that here. This is where we will start our class today, and see you soon.